All right. Hi, everyone. This is Grant again. Uh, there was seemingly a good response to my FoxP3 video, so I decided I'd stick with it and make a couple more molecular bio videos just for the final for you all to look at and get my chaotic explanations with my poor handwriting and bad microphone. So that's kind of fun. Today, I'll just be talking about epigenetics fundamentals. This is really foundational to a lot of stuff we learned around the final, and it's important to get a good grasp on it. So before we even get started, it's like, what is epigenetics in general? I was trying to explain this concept to my wife, she's a high school biology teacher. So we both do biology, it's just at different levels and to different audiences. And I was like, well, in high school, genetics is so simple. You do your Punnett squares, let's say for hair color, brown and blonde. You do your Punnett squares, you see what works out, you understand, oh, heterozygous, homozygous, dominant, recessive. And obviously, hair can either be blonde or brown. Except, it's not actually really that case in real life. So in real life, I have hair on my chest, but not hair on my back. I have hair on my head, but not hair on my teeth, even though it's all the same cells, right? So why is my hair different texture in some places? Why is it different colors in some places? Why don't I have hair all over my body on all of my cells? And a large part, it's just epigenetics. It's your body. Every cell has this whole, you know, giant genome with tons of genes. But obviously, it would be pretty silly to express all your genes all the time. So let's say the top of my head, the cells, my hair follicles have to be like, oh, we have to make lots of relatively straight brown hair. Okay, well, let's just say, what if there's only one region? Let's say this is the straight brown hair region. It's like, well, how do we'll make this a blonde region in my genome. Well, how do you magnify, like, how is the hair on just the top of my head only brown and not blonde and everything else? So that's because your body, through epigenetics, just has various mechanisms of promoting some genes. So make sure my brown straight hair grows using those genes and suppressing other genes. So I don't have blonde hairs. I don't have teeth growing out of the top of my head because all my tooth genes are suppressed through epigenetics. And that's pretty cool. Unfortunately, going down more and more complicated. Come back here. Epigenetics is kind of complicated, but don't worry, we'll get through it together. So there's sort of three big mechanisms of epigenetics. There's histone modification, DNA methylation, and non-coding RNAs. And we'll just talk briefly, very bare bones understanding of all of these. So histone modification, seemingly this is one of the more common. A lot of times when you see stuff about epigenetics, this and especially DNA methylation are definitely your most common you talk about. So to get started, here's let's call this a histone. So this is this ball. Get my color out. Histone. So most of you should know this by now, but a histone is basically it's an octamer. It's like 8 peptides put together to form this giant ball that your DNA, which is my little rainbow thing wrapping around, that your DNA binds around. And in the most simple form, your DNA, right? So normally you get your double helix, right? Well, that's convenient for storage, but it's not super convenient for storage. We have a lot of DNA. So how do we pack it tightly? Well, I'll wrap my DNA around my histone like this, and it's compacted into something called chromatin. So I'll write that out. Chromatin. Ooh, it's lagging behind a little bit. Okay, so chromatin is just compacted DNA. So you have your double helix DNA, you squish it up even closer together. It's called chromatin. Well, we can pack and unpack that chromatin even more. So around the histone, you may have some regions like up here where it's pretty tight to the histone. So you can see, here's my histone, here's the little arms coming off of it. Oh, the DNA is actually bound pretty close. And by the way, this is not a realistic model. This is, this is a helpful way to think about histones and DNA. So here, oh, it's bound pretty tightly to the histone. So this, it's tightly bound, you would call heterochromatin, which just means it's well packed. It's hard to get to. It's in cold storage. This is. So if we go back to my mustache or my hair example, right? I don't have blonde hairs very much on the top of my head, but I do in other parts of my body. So here, let's say, this is the blonde hair region. I highlighted here. Well, obviously that's in cold storage. I don't need that in my hair follicles on the top of my head because I'm not blonde. So it's tightly packed heterochromatin. Now, the opposite of hetero, and I'll write that out. We'll make that easy. Heterochromatin. You gotta forgive my handwriting here. I'm getting used to writing on the Microsoft whiteboard. Now, let's go down here. Oh, look, it's not really very tight to the histone. So I'll call this euchromatin. So let's say this is my brown hair region and the hair follicles on the top of my head. It's super far away. And U just means good. So this is like good chromatin. This is nice and wide open. It's easy to access. So you may ask, what do you mean easy to access? Like it's all DNA, it just copies, right? Well, that's not really the case. It's like you may have noticed in molecular biology, everything is just shapes moving around, touching each other, right? So an easy way to think about it is you have your RNA polymerase is a shape. RNA pol. It's a shape, it's an enzyme, and guess what? I have to get to the DNA, which is wrapped around these histones. Well, it's a whole lot easier if I land on this euchromatin with not a lot of steric hindrance than if my RNA polymerase tries to go to this heterochromatin. Oh, it's too tight. It's too closely packed together. My big old enzyme can't actually get in there and do anything. And so, really, it's just a way. Changing the histones is a way to either make it so things are more expressed or not expressed, sort of by making it heterochromatin or euchromatin. And even a little bit deeper, there's basically 4 mechanisms of this. So your first one is acetylation. So let me clean up my model over here. Do some erasing. All right, acetylation. That's the first one. So for those of you who are chemistry oriented, an acetyl group, it's just a little chemical thing like that. You'll just add it to sort of these tails of the chromosome. And in a way oversimplified version, it turns heterochromatin, okay, which at the end of this tail, I have my heterochromatin. I add a little acetyl group at the end, and then whoosh, the steric hindrance pushes out my chromatin, changing it to euchromatin. It's less tightly bound, basically because I've wedged an acetyl group in there. To make it looser, oh look, RNA polymerase can bind better. And so acetylation, as you might remember if you watched my first one, is done by 2 enzymes. So actually my favorite is, you have an acetyl transferase. We'll just call it ATase. So basically take an acetyl group, plop it on there. Things are nice and happy. Well, sometimes you have to turn things on and off. So acetylation comes with a complementary deacetylation with my favorite HDAC, which is histone deacetylase. So HDAC sort of does the opposite. Say, acetyl group, I have to turn that off. I'm tired of growing hair. I'm getting old. I don't want to grow any more hairs. I'm going to deacetylate and tighten this chromatin up. And HDAC will come over and launch away at that acetyl group. And this is basically a process. They're fighting each other all the time. It's constantly happening. Just the conditions of your body will determine, oh, well, do I have more acetyltransferase? Do I have more HDAC? Uh, how loose is my chromatin? How many acetyl groups do I have? So it's very briefly acetylation. So acetylation is always making euchromatin of heterochromatin, and then deacetylation is sort of the reverse of that, using histone deacetylase. Now, a little more complicated is methylation here. So methylation, you're just adding another chemical group. It's just a methyl group. At the end, this can have multiple effects. So let me reference my thing just to make sure I'm doing it right. So methylation, I'm adding a methyl group here. So okay, well, where is my methyl group? Is it, going to be like acetylation where I put it at the tip of these little arms? Oh, there's more steric hindrance. The DNA can't bind as close. Or let's say there's a region of DNA or let's say there's a region of the histone that's really, I just want to make it harder for things to get in there. Maybe I can do tons of methylation and then boom, lock it up. It's all steric hindrance in different ways. But the important thing to remember is methylation can go both ways. It can enhance or it can restrict. So we'll write that down. So we could say enhance or restrict. You can see, yeah, it's there. Stuff that counts. So acetylation is just always enhancing. Okay, and then these other ones, similar mechanisms. So we'll talk about phosphorylation. So even though phosphorylation sounds big and complicated, it's basically the same stuff. So phosphorylation, you might notice it's just named for what group you're putting on it. So phosphorylation, we have a giant, phosphate. Just call that P. It's usually how you draw a phosphate. Your phosphorylation is just another way. This is effectively super negative. And if you're, oh, well, DNA has a phosphate backbone, that's also negative. So here, it's not necessarily steric hindrance. Like in methylation and acetylation, this is like, basically forcing space. So it's pushing away that negative DNA, opening it up, turning it into euchromatin, just based off the negative charge rather than any structure inherent to the phosphate. Sort of a negative force field around making the chromatin bulge out, be nice and accessible to RNA polymerase. And then lastly, you have ubiquitation, which is just, it's a protein called ubiquitin. So I'll draw out my little ubiquitin, something like that. And ubiquitin, well, it basically does the same thing. This, instead of being, a molecule getting added or a chemical group getting added to the end of it, it's basically just so it's not causing structural steric hindrance like in acetylation methylation. It's not doing this negative force field propelling it away like in phosphorylation. It's just an actual protein, ubiquitin, that will come over and just change how accessible the chromatin is directly. So really, it's the same thing. It's how close is the chromatin to the histone? Is it euchromatin? Is it heterochromatin? Can the RNA polymerase get to it? Can it not? And that's basically the fundamentals. So you just need to know these four things do it in different ways. And yeah, lots of cool options with histone modification. So another pretty simple one here is DNA methylation. So we talked about histone methylation. It can enhance it or it can repress it based off of where you put it on the histone. This is the exact same concept here. So let's say I want to methylate some DNA. So let's say I want to make it so my DNA can be super easily accessible. So I am going to go in, I'm going to add a bunch of methyl groups basically on top of my gene of interest. And so what that does, it's the same thing. It opens it up, right? So here, instead of modifying the histone, I'm modifying the DNA directly. They say, okay, well, I want my gene of interest to be more accessible, so I'm going to methylate on the gene or around the gene to be like, okay, I'm here, I'm super accessible. RNA polymerase, you have all the real estate in the world to come and bind to me. So say, oh, look, it's a nice clear field. I'm going to bind to the promoter and we're going to code. Well, that's all great. However, there's other ways of methylating. So if you methylate on the gene, you open it up, it's more accessible to RNA polymerase. But let's say I want to block something. Well, conveniently, I can also go and I can methylate the promoter. So again, it's the same methyl groups. It's the same process. It's just, with a methyl transferase. So I'll call this MTACE. Methyl transferase comes in. There's a bunch of methyl groups on the promoter. And then, RNA polymerase, oh, look, I'm going to land on this promoter and encode it. But there's a giant roadblock. Man, can't do it. So it's just important to remember DNA methylation. This happens all the time. So you'll have de novo methylation. De novo. Which basically means, okay, well, once upon a time, I was a sperm and an egg, and they met together and did all sorts of recombination to make Grant and differentiate my cells. And during that process, methyltransferase is working overtime, saying, well, I want this cell to be this, that cell to be that, putting methyl groups on promoters, putting methyl groups on genes to begin with, and that's all great. Which seems, oh, okay, that's great until, me, Grant, a one cell at that point, I have to, divide into multiple cells. There's a lot of cells that make up a Grant. So do I have to go through this process all the time of methylating? And the answer is yes. So it's just when it first happens in super early cell development, it's de novo. It's the first time you're being methylated. It's basically, you're shuffling things randomly. So maybe an alternate Grant, I'm methylated slightly different. I have blonde hairs on my head and I have a brown mustache. Today, now I have brown hairs on my head and I have sort of a multicolored mustache. And it's basically just a quirk of de novo methylation. So here's me at the beginning. So, okay, well, we've shuffled the cards around, and Grant is going to have brown straight hair and a multicolored mustache. Well, obviously, consistency helps. I'm not going to reshuffle every single time. So every time my cell divides and I replicate my DNA, I'm basically just going to copy all the methylation already there. So if I'm suppressing things, if I don't want blonde hairs on my head, my blonde hair promoter is super methylated. It's like a roadblock in the way. If I want brown hair, I'd methylate my brown hair gene on top of my head, all my hair follicle cells. Okay, well, we want more of this. This is good. So just important to know, DNA methylation goes both ways. It's basically randomly shuffled at first in de novo, and then whatever your random assortment is stays basically the same throughout your life as maintenance DNA methylation. It's just copying how your de novo shuffled it. So slightly more complicated, sort of a third mechanism of epigenetics, are non-coding RNAs. So RNAs do a lot. This is like, the frontier of molecular biology is RNA, seemingly. There's all sorts of cool stuff with RNA. And I mean, there's a bunch of different RNAs that don't encode. So hopefully this should be review from a previous exam for you guys, but we covered microRNAs and siRNAs. So microRNAs, they're both. So they both basically follow. Okay, here's a super small RNA sequence. It's like 20 to 23 ish nucleotides. So it's super small. It doesn't code for anything. Then you'll form your dicer, or not your dicer, your RISC complex, and you'll go and silence a bunch of messenger RNAs that way. mRNA. RNA. So microRNAs and siRNAs, we covered this previously. This is a review. I'm not really going to talk about it. This is just reminding you that these are small. It's either a microRNA, it's an miRNA, or a small inhibitory RNA, or a small interfering RNA, and they're pretty similar. So I should say, here we go. siRNAs have this risk mechanism, right? miRNAs don't bind to risk, but they're there gunking up the works. So they're basically the same thing. That's the slight difference. Is an siRNA is attached to a risk, an miRNA isn't. It's just there to sort of attach to things and mess it up, and then the cell's like, I don't want double-stranded RNA, cut it out. So again, that should be review. So really, the meat and potatoes of it is you have Pee-wee interacting RNAs or piRNAs and long non-coding RNAs. So we'll talk about piRNAs really quick. So a piRNA, again, this is super small. This is like 24 to 31 nucleotides. So pretty small. And this is here to silence transposable elements. So if you remember what a transposon is, say, here's my DNA. I'm drawing circular, like obviously humans don't have circular genomes. It's just, force of habit from bacteria, which is most of my work. And let's say, let's make a transposon red, right? So transposon, it's also called TN. It's a jumping gene, right? So it's got basically mirrored DNA sequences on each side, and it's really good at picking itself up, moving elsewhere in the cell, keeps going all the time. Well, that kind of sucks if you need to have like a germ cell. Like, I don't want anything messing with this. Like, I don't want to have super weird kids with genetic abnormalities because there's transposons going everywhere, right? That would be not conducive to the survival of the human race, right? So you have the Pee-wee interacting RNAs. So effectively, what this will happen, here's my little piRNA. These will be relatively specific to a transposon. Go over, bind to the transposon in my germ cells. So I should specify that. Germ cells, things where you want super high stability. Don't want any complications here. Germ cells. So that'll basically silence these transposons. So, my piRNA went, attached itself to this transposon. That transposon's not going anywhere. It's locked in place, and that's great because I don't want it moving around, causing damage, interrupting genes. Got to keep it consistent. So it's relatively simple. And then, while all of these are super small RNA fragments, we also have the opposite, these long non-coding RNAs. So while the rest of these are small non-coding, we also have our long non-coding, which is huge. These are like more than 200 nucleotides. So pretty big by comparison, right? Well, these basically assist in chromatin modifications, right? So they're kind of like bridges. Like, like if I have an enzyme sort of going to work on my chromatin, sometimes I just need some general structure. Like let's say my ubiquitin wants to come in. Sometimes I'll need some scaffolding to fit proteins, right? And you can use long non-coding RNAs just as structural support and modification that way. And it can also, it can help recruit extra proteins. It can help block proteins. It's basically all based off the shape. These are like bridges. I'll call it a Call it a chromatin bridge. Chromatin bridge. Except it's some cursed, you know, it's a bridge and a reverse bridge. Where, okay, here we go. Let's say a bridge crossing a river, great, helps me cross the river. A bridge built, basically on top of the road I'm going, it's not helpful. Like let's say here's my road, dotted line, great. Well, what if I built a bridge actually just not even above it, directly on top of the road? Well, it's really inconvenient because my car can't, drive through a bridge unless I like ramp over it. It would be really annoying. This is basically what long non-coding RNAs do. So if the road is chromatin, I'm either making it easier to access, I'm making it harder to access, just by building bridges. Sometimes in great places, sometimes in really inconvenient places. It's just whatever matters the most. An example of these are exist RNAs. Exist RNA. So let's say, sorry, my notifications came up. Okay, exist RNA. So you have X chromosome, right? Well, let's say you're female. Two X chromosomes, right? Well, you don't. It's kind of redundant having two. Like, are you going to encode everything on both X chromosomes? It's like, no, it's not the case. So I'm going to take one chromosome, and I'm just going to put long non-coding RNAs all over it. I don't need it. So let's say, oh, well, I just have my dad's X chromosome, or I just have my mom's X chromosome, because it would be redundant if you're encoding everything on both X chromosomes all the time. And so I'll just take one of them and have my exist RNA, which are these long non-coding RNAs, and I'll just cover one up, silencing most of the X chromosome. And